Hey everybody, and welcome to the lecture on the auditory and the vestibular system. We will spend most of the lecture today on the auditory system with just a little bit of time dedicated to the vestibular system. There are only a few concepts there to cover, but it's very important that you also pay attention to those. So let's talk first about sound as a stimulus, because we need to know what the energy that we're transducing is. If you remember the beginning of the visual system lecture, we talked about the different systems, of sensory systems specifically in the brain, being responsive to certain kinds of energy from the environment. Starting at the most basic level, audition is our sense of hearing. And we use this system for a few things. The first of these is probably the most obvious. We use this to detect sound. So being able to just hear that something has occurred around us is the primary goal of this system. Even beyond that, though, we need to be able to localize where sounds come from. It's very important for us to use this. And we talked a lot at the end of the visual system about how information from all of our sensory systems comes together. And then the last thing is we need to be able to identify and differentiate sounds. It's fairly important for you to be able to use your auditory system to identify certain things, because being able to identify things that are dangerous by their sound gives us an extra advantage for things that are coming from behind us, or things like that that allow us to protect ourselves. So the way that sound is created is through vibrations in the air, and there are actually a couple of components to this. So what we have is that air molecules will alternate between two different points. So we call this either compression and expansion, or it's sometimes called compression and rarefaction. And this alternation between these two is actually what the ear is transducing. And so a lot of times we see sound as kind of sine waves or things like this. We see it as either sine waves or very complicated waves. And this is what's happening. This is the signal that the ear is getting, is exactly what's happening in the air right here. This kind of push and pull, because when the air molecules group together, they create pressure. And when they expand, they create something you could think of, at least to simplify it, like a vacuum. And if you've ever looked at a speaker that's playing a sound, you'll see that this is kind of exactly what it does, right? There's times where it pushes forward, and there's times where it kind of sucks back. And so what's happening there is it's creating that movement of the air by creating the vibrations that match what's being plugged into it. Another way that I like to tell people to think about this, at least the idea of compression and rarefaction, is if you think about what happens when you throw a stone into a pool of water, it's not that there's just one big giant wave going out, but instead you end up with ripples. And the ripples are basically alternations between the water being compressed and it being drawn in at different spatial increments. So all of these things have kind of a multi-part component to them of compression and rarefaction. So for a long time, I had been coming up with good examples of people and their ridiculous car stereo systems to show you a little bit about vibration and sound. And usually it involved somebody's windshield getting shattered or something like that. But a few semesters ago, I found what I think is the best example, and I've been using it ever since. So I'm going to warn you ahead of time. If your speakers or your headphones are on really loud right now, you might want to turn them down a little bit. But I want you to see just exactly what these compressions and rarefactions and this big vibration of sound actually can look like. I don't recommend doing this for your car, but I do recommend it for your entertainment. You're gonna have to wait a second. So I don't know about any of you, but I had never seen a door or a window bend before. Kind of crazy. And those bends and movements happening at different frequencies, those ones very low and others very high, are what give us the qualities of sound. So what we heard that was bending the window was mainly things that are a low frequency, but we also were hearing things that were high amplitude, and in some cases with complex bass lines that have more complexity to them, kind of a lot of different features to them, especially as we hear the rattling of the car and the speakers and all the things all at once. So we have multiple features of sound that we care about. Breaking down each of these elements a little bit more specifically, if we look at frequency, then this is determined by the number of cycles of compression and expansion per second. And when we talk about cycles per second, the unit that we use for this is hertz. So when you saw that window and you could see the ripples in it, you could actually see those frequencies because they're so slow, right? There's so few compressions each second that you can actually see them, whereas something with a really high frequency would vibrate very, very quickly. And if it's vibrating at 1,000 times a second, it would be very hard for you to see something vibrating that quickly. We as humans can perceive things between about 20 hertz, so 20 cycles per second, all the way up to 20,000. And that's just kind of a standard bit of the range, but something you should notice is that we don't perceive all of these equally. So the loudness that we need to perceive something that's at 20 hertz or 20 kilohertz, 20,000 hertz, is very different. So there's some kind of drop-offs there. But the things that we care the most about, like language and music, happen to fall into the range where we're able to hear at really low values of loudness. So, and that unit of loudness we call a decibel. So this is kind of the way that it shows you that the system is designed for the things that, again, we need and use the most, but we're able to detect things that maybe fall far outside that range as well. The loudness of a sound, which is that unit decibels that I just mentioned, is determined by how different these compressions and expansions are from each other. So something that's really loud will cause kind of strong compressions followed by strong expansion or rarefaction. And this creates, at least in the way that we would look at this as a sound wave, a larger amplitude, so a bigger height. And you could measure this from the bottom to the top, or you can measure it from the center line because it's kind of symmetrical to the peak. Doesn't really matter which one. But that difference between how much compression there is and how much expansion there is what determines amplitude. As we already said, the unit for this is decibels. The decibel system is a system that happens to be logarithmic, so it's a non-normal scale, and that means that as things go up 10 decibels, they actually get louder very, very quickly. It doesn't go in a linear count system, and they get exponentially louder at each 10 decibels that we go up. And again, we visualize the decibels, if we're looking at a waveform like this, as this amplitude or peak of the waveform. Timbre is probably the most difficult to talk about because it's a little bit difficult to wrap your head around. So we define timbre as being the quality of a sound. And we could have similar sounds like you see here, a tuning fork, a flute, a voice, and a violin, and they could have the same frequency and the same loudness, but we would perceive them as different due to kind of the different effects of the sound. And so even starting with the tuning fork, I want to just point out, so this is probably one of the closest things we would ever get to a sine wave. It's something that you hold and it vibrates in a very precise manner because it's made of metal. The vibrations are pretty close to a pure vibration and not even quite that. However, take something like a violin playing the exact same frequency. there are actually a lot of kind of little nuanced bits of complexity there, and that's what creates the quality of timbre. So how that sound then vibrates through the wood, how the person's hand might subtly change their placement on the string, how the speed of the bow moving across the string changes the sound over time just ever so slightly. And all of those things come together to create a much more complex sound. So there's some instruments that are much more precise and others that we love in part because they have this quality to them, this timbre that is beautiful based on all of the different complexity in the instrument. So we tend to think of this, or at least a good way to think about this is that there's kind of a primary frequency and that the timbre is made up of all of these subtle, nuanced other frequencies coming together. We wouldn't be able to get away with talking about audition without talking a little bit about the anatomy of the ear. And the most important thing is that you understand how we take these compressions and rarefactions and turn them into a neural signal. And understanding the way that the ear is set up is important. Starting from the most basic level, there are three parts of the ear. So the outer ear is primarily composed of the pinna, or what you will see as the ears on the side of somebody's head in the ear canal. The middle ear has a tympanic membrane, as well as the three bones of the ear, the malleus, the incus, and the stapes. And the inner ear, the primary piece that we care about here is the cochlea, which is the primary structure for transducing sounds and signaling what those sounds were to the brain. So in terms of walking through this process, sound would be funneled into the inner ear through the pinna. And there's some really important features that dictate why our ears are shaped the way that they are, right? We could just have a hole in the side of the head, and some animals kind of do have that, but having this pinna that has these funny shapes and other things to it gives us some advantages we'll get into later. Sound travels through the ear canal, and then it reaches and it creates vibrations on the tympanic membrane. So all of those compressions and rarefactions that we had just seen are going to come in and they're going to push the tympanic membrane in and pull it out. So this is creating a vibration, not unlike somebody jumping back and forth on a trampoline. Another way to think about this is going back to the Latin root for tympanum, which means drum. So you could think about somebody hitting the head of the drum with these vibrations. In the middle ear, we have these three bones, and collectively we call them the ossicles, and they're important for transducing sound. So the vibration on the tympanic membrane is going to be passed through these bones, which take it and turn it into kind of a mechanical push and pull on the cochlea, especially through this oval window that you see here. So the first of these is the malleus, the second is the incus, and the third is the stapes, mainly named for their shapes. So the malleus looks like a hammer, the incus looks like an anvil, and the stapes, it looks like kind of a stirrup, just based off of the original descriptions of them. And again, these bones are responsible for taking the signal from the tympanic membrane and basically transmitting it into the cochlea. So what happens here is that we're taking vibration and we're actually turning it into fluid pressure in the cochlea. So the cochlea is filled with fluid, and when the tympanic membrane vibrates, it causes these bones to move, and they push at the stapes in and out along with the vibration, and this is what causes kind of pressure changes in the cochlea. So some of you in the class come from very diverse backgrounds. We had some people in engineering and other things like this, and the primary reason for this is that there's a major advantage to having kind of a fluid or a hydraulic system. The best example that I can give for all of you are the brakes in your car, right? If you, a human being of average shape and size, were to try to stop a car by pressing on a pedal, you might as well be trying to play Fred Flintstone and putting your feet on the ground and trying to stop the entire vehicle. And so instead, what we use is a system that's a hydraulic system where you press and compress a fluid. And many of you probably remember from physics classes and other things like that, that fluids don't compress very, very well. So when we do create pressure in fluid, it's high efficiency. And that creates a big advantage for taking something like vibrations in the air, which may not be all that precise, and turning them into a very precise signal within the cochlea that's very easy to distinguish. And part of the advantage of this is that you can take multiple complex frequencies and you can transmit them all at the same time by creating kind of complicated movements in fluid, not unlike that ripple in the water that I showed you a few slides ago. The cochlea is divided into three separate sections, the scala vestibuli, the scala media, and the scala tympani. We'll talk a little bit about the importance of these. But I say this because we're referencing the anatomy, not necessarily because it's something you need to know just for the sake of knowing it, but their functions will become more important. So when this fluid pressure comes in, the thing that you need to know the most is that there is basically a receptive part of the cochlea. We call this the organ of Corti, and this is where the meat and potatoes are for what you need to know in terms of physiological psychology. So if you look at these channels here, right, we've cut a hole in the cochlea and we're looking through. Then what you can imagine is that this scala vestibuli and the scala tympani are going to be filled with fluid, and basically as vibration comes from the outside, the fluid in here is going to push and pull. And this push and pull affects different parts of this organ of Corti differently. So the bottom part here is called the basilar membrane. We have hair cells in the middle that are anchored into this basilar membrane. And then at the top here, we have a tectorial membrane. Okay. So keep in mind this idea of vibration and fluid changes, and then think of all these three pieces. So what happens is that when we have these changes in fluid inside of the cochlea, then they cause this tectorial membrane to move relative to the basilar membrane. So I want you to see this. This tectorial membrane is kind of hanging here. So when we have movement of fluid here and pressure that builds up, if the pressure builds up a lot, it's going to cause this entire channel to expand and bend, and that causes this tectorial membrane to move back and forth. And when it does that, these hair cells get pushed back and forth as well. So this causes movement. And these hair cells are really the primary cells doing all of the transduction. So much like in the visual system, when light hits, in this case, we have a kind of a mechanical sensation that pushes these sensory neurons back and forth. And this is what produces receptor potentials. So remember, we're not talking about action potentials. We're talking about basically changes, depolarizations or hyperpolarizations, depending on how these hair cells get moved. So an easy way to think about this, for all of you that want to do an experiment just before we get into this whole process of auditory transduction, is if it's very easy for us to feel things by touch. So if you take your hands and you just rub them back and forth, for those of you that are Karate Kid fans, you can think of this as kind of the Mr. Miyagi move. And that movement back and forth is creating changes in pressure at a given spot on your hand. And so that back and forth movement, maybe you can hear me doing it, is exactly what's happening between that tectorial membrane and the basilar membrane. It's just they're moving relative to each other. So then let's talk about the signals coming to and from the cochlea. So we're going to start a little bit backwards, and we're going to be talking about inputs that come into the cochlea. So the cochlea, as we've said, is kind of this primary auditory sense organ. And there are some cases where we need the cochlea to get signals from the brain. So one of the best examples of this besides just the one I've given here in the slide, is that, you need to habituate to a constant noise. And so if there's a constant, humming from the air conditioner in your house or something like that, you get used to it after a while, and your brain basically says, okay, turn down the volume on this humming sound. But the other thing is it can prepare us for particularly loud noises. So when you yourself are getting ready to scream, your brain will actually send a signal down to the cochlea that says, hey, I'm gonna, let out a really big scream. You should probably get ready for this. And so these are the kind of signals that we send down from the brain. And these signals come from a part of the brainstem, specifically called the superior olivary complex. And these signals can come down and synapse right on the hair cells. So a good example that many of you will probably remember is, or at least another way to conceptualize this, is when we talked about, kind of picking up that hot bowl and inhibiting the motor reflex, and just talking about how, kind of the bigger picture of how action potentials work and how signals can come up, but we also can cancel them with inhibitory signals. That's what can happen in the case of the cochlea here. In terms of the output, the information coming from the cochlea works very similar to the information coming from the visual system. We have these hair cells in the organ of Corti, which again is the basilar membrane, these hair cells, which are the entire cell here, not just these little hair-like protrusions, and then the tectorial membrane. And we have then the cochlear nerve, which is created by a bundle of axons that come from bipolar neurons. So on one side, they receive the transmission of signals from these hair cells, and much like in the visual system, this input is from the neurotransmitter glutamate. And then glutamate can trigger action potentials via these cochlear nerve ganglion cells. And these are the signals that end up going on into the brainstem and onto auditory cortex. And we'll talk about that a little bit more. What's important to know here is that the way that we get different frequencies depends on what part of the basilar membrane bends because of a given sound. So the basilar membrane, of course, is being affected by pressure, but the important thing is that different frequencies affect different parts of this basilar membrane differently. So if we were to take the cochlea, which kind of looks like a snail, and stretch it out, then this length of the cochlea creates what we think of as a tonotopic map with different frequencies spread across. Stepping back again just for a second from the idea of frequency, the way that all of the frequencies are transduced is exactly the same. So we've said we have a map across the cochlea, and what's important is the bending of this tectorial membrane and basilar membrane relative to each other, because that causes changes in what these hair cells do. So what ends up happening in each of these cases is that these little hair-like protrusions at the end of the hair cells are called cilia. And they actually are the thing that becomes used to generate a rate code in the auditory system. So they have a preferred direction where if they are bent from the tallest towards the smallest, then that actually shuts them down, and that will eventually create a low rate of firing. Whereas if they're bent from the smallest towards the tallest, then that's their preferred direction and creates a high rate of firing. And all of this has to do with some hair-like threads that actually kind of connect these tall and small cilia called tip links. So what happens is that these tip links that we're talking about are actually connected between a small cilia and a large cilia. And the reason that this matters is because their job is to actually control a channel, an ion channel that's up here on the taller tip links. So when they bend from tallest to smallest, then that actually creates slack in this tip link, and the channel is closed, and that'll create very little neurotransmitter release and a low rate of firing. Whereas when they bend towards the taller of the two, then it causes this tip link to become taut, and it opens up this channel here and causes a higher rate of firing. Unlike what we saw in neurons, the primary ions here are potassium and calcium, but potassium is by far the more important of these two for driving this process here. And this ion channel up here, just like I was saying, you could think of this as some sort of like a pulley or a stretching. You can think of this as like a tripwire, because the channels that we use to open these are called trip channels. So this pulling here kind of opens this trip trap and allows the potassium and calcium to enter. And when that happens, then it depolarizes the hair cell that these cilia are attached to and causes it to release more neurotransmitter, and then of course causes a higher rate of firing in the bipolar cells that are receiving that neurotransmitter, in this case, this glutamate release. So this brings us to something really important that we need to talk about in the way that the auditory system functions. And this sometimes takes people a couple of passes to get. So there are two types of coding that can happen within the auditory system. The first of these is place coding. And so I'm going to start with this and go backwards first, because I mentioned it a second ago. This is the idea that frequency is coded by the location along the basilar membrane where those hair cells and the cilia and the tip links are being bent and activated. And this means different parts of the basilar membrane respond to different frequencies. The other type of coding is a rate coding. And this is a type of coding that is mainly used for low frequencies. And it's coded for by the rate of firing in a certain subset of neurons from the auditory system. So let's talk about this rate coding for a second. Very, very low frequency sounds. The thing that I'm going to say that I think will help make this make sense is that our cochlea is not really designed to detect some very, very, very low frequency sounds. And instead, what they do is cause vibration. And the louder that they are, the better they are at kind of poorly activating the frequencies we are tuned to detect. So low frequencies cause the apical end, which is kind of the tail of the cochlea, to move, and basically causes this tail to kind of waggle and vibrate back and forth. And what we've seen is that neurons can fire in synchronously with this. So as the cochlea kind of vibrates, then we'll get neurons to fire along with the peak of that vibration. And then the louder the sound is, the more neurons that it will recruit. And part of this is because we talked about neurons being tuned, right? We talked in the visual system about neurons being tuned. And when something is poorly tuned, or if a neuron is poorly tuned to whatever you're giving it, let's say, a line of the wrong angle, using the visual system example, it doesn't mean that it'll fire no action potentials, it'll just fire fewer. And so we think these low frequency sounds that our cochlea is poorly tuned to, as they get louder, they can still cause some of these neurons to fire, or to some of these hair cells to activate and cause these bipolar cells to fire action potentials. And so this is kind of a case where we know how this system works, but it's actually the exception. Only about 5% of frequencies that we perceive are encoded in this using this rate code. Which should lead you to a question like, why don't we encode every frequency using a rate code, right? It could be the case that we had an auditory system, and if a sound were vibrating 20 times a second, I could have a neuron that fired 20 times a second. And if a sound were vibrating 100 times a second, I could have a neuron firing 100 times a second. I could have neurons that were tuned to how fast the sound was vibrating, right? Well, it turns out that that's not the case, and this brings us to something that if we were in an in-person class, I would ask you to guess at how quickly neurons fire, but we don't have that luxury, and so I'll cheat and just give you the answer this time around. Give or take maybe a little bit, the very fastest neurons in the brain for short bursts probably fire on the order of 100, maybe 200 times a second in short bursts. And they wouldn't maintain that. They wouldn't probably even maintain that for a whole second. Some of them can fire, very rapid bursts for a short window of time. Think of this like a cheetah sprinting. It can sprint very fast, but for a short window. And that's a very small subset of neurons. Most, and I've recorded neurons in many parts of the brain, only are able to fire maybe at 20 or 30 times a second. That's probably more like the average is 10, 20, or 30, somewhere in that range. What this means is that we can't keep up with our hearing, which can go up to 20,000 hertz or 20,000 times a second. So for the other 95% of the sounds that we hear, we use what we call the place principle. And the idea here is that basically the different locations of hair cells along the basilar membrane determine what frequency they respond to. So you can see the middle ear would be here, and as we get into the cochlea, which is again stretched out, so you can see its entire extent here, going from kind of what we think of as the basilar end, where the high frequencies are represented, all the way down to the apical end, where lower frequencies are represented. The reason that this works is that the basilar membrane up here, right at the beginning of the cochlea, is very thin, and so it can respond to high frequency sounds really easily, but it responds kind of poorly to low frequency sounds. And on the other end, the basilar membrane starts to get thick, and it responds better to low frequency sounds, again right near this apical end. So this means that different frequencies all across this range bend hair cells along different portions of the basilar membrane. And again, what you'll see is that part of this map involves the frequencies that we care about the most being kind of represented by more total space along the basilar membrane of the cochlea, and really, really high and really, really low frequencies don't get nearly as much representation in space. So let's now take a look at these two ideas again of a rate coding versus a place coding with a picture in front of us. So the lowest frequencies of sound are represented by recruiting more neurons towards this apical end of the basilar membrane. And so the way that we get this rate code here listed as frequency is that instead of having 1 or 2 hair cells recruited, the louder the sound is, the more cells that I recruit. So a weak sound might only recruit a few, and then as the sound gets louder and louder, we start to recruit more cells. And this is what allows us to perceive really low frequencies. So this is things that are below 200 cycles per second, which if you go onto YouTube, you can usually get examples of all kinds of different pure tones. So if you want to hear what a 200 hertz tone sounds like, then you could probably find one. But I'll tell you that even this is probably a bit of an overrepresentation. It's probably things that are more like below 30 hertz that are really, really represented using this kind of a rate code. For all other frequencies, we use a place code, which means that there's only one cell in one place on the basilar membrane that is tuned to that frequency. So that means that while this low frequency might have recruited these 4 different hair cells at 4 different places, higher frequencies from 30 to 20,000 hertz are only going to recruit one set of hair cells from one spot on the basilar membrane. Another thing that's important is that the action potentials, if you think of each of these spikes here as being an action potential, are going to fire in phase with the frequency of that sound. So they're going to fire at the point of a compression, which makes sense if you think about what we just talked about with the hair cells, right? So when this part of the basilar membrane is compressed, it's going to cause the hair cells cilia to bend, and it's going to cause glutamate release, and then you're going to have action potentials that are in sync with the point at which that bending happens, right? So that's in phase with these compressions of the sound. Some really good evidence for how the cochlea does this is supported by the use of cochlear implants. So for those of you that don't know how this works, typically there is kind of a microphone piece that is next to the head externally, and then there's an internal implant that includes a sound processor, and what's implanted is actually a series of electrodes. So we talked about different ways of electrically stimulating neurons and things like this, or optogenetically stimulating neurons, but we know that we can use we can activate neurons. And so this internal implant is basically a long wire with a series of electrodes that are fed into the cochlea, and they get wrapped around the entire length of the cochlea. And they have different, you can see all maybe these little different dots on this implant. Those are each different electrode locations. And what this sound processor does is it takes sound and it recreates the different frequencies and only stimulates the part of the cochlea that correspond to the different frequencies of sound that are heard by the microphone that's picked up. So this is a really cool way that we can show this place coding that we see inside the cochlea, is that the fact that cochlear implants work and are able to recreate the perception of sound just by activating these neurons in somebody that might have a congenital hearing defect or something like that is really strong evidence for the way that this entire system works. So to give you a picture again of how that would work is that we start out with the basilar membrane. We have different frequencies represented across. And so if we were to have stimulation at specific points, if I stimulate with an electrode here, that might stimulate at 1600 hertz. And if I stimulate here, it's going to stimulate a set of neurons that would normally represent that basilar membrane bending for something like 25 hertz. And so you can recreate this entire range of frequencies just as if it were playback through a speaker or something like that. Another important thing that kind of goes along with our localizationism or functionalism principles that we've talked about is that this map that we see in the basilar membrane corresponds perfectly to a similar map in the auditory cortex. So specific regions of the basilar membrane that represent certain frequencies then project into the primary auditory cortex, which maintains this same kind of a tonotopic map. So much like the visual system, this mapping, this systemic mapping is maintained across the entire system. It's an organized system. This brings us to the auditory pathway. So we already said signals that are transduced by these hair cells, they travel from the cochlear nerve, and that they go to the brainstem. And the place that they arrive in the brainstem is called the cochlear nucleus. And it's on the ipsilateral or same side of the head as the ear would be in this case. And then most signals are going to cross the midline to the opposite side of the brainstem, right? Which we also call the contralateral side, to a part of the brainstem called the superior olivary complex. And then this signal passes through kind of a bundle or group of grouping of axons, we call the lateral lemniscus, to a nucleus in the midbrain called the inferior colliculus. And one thing that I want you to notice here, which is going to become important for some of the somatosensory systems and things like that, is that there is kind of a second fast track here, where some projections skip passing into the superior olivary complex and they go straight to the inferior colliculus. And so one thing that I'll say is that the example I always give is when we have certain pieces of information that sometimes need to get fast-tracked. And what we saw in the visual system is that each nucleus, you could think of, when things went through different layers or connections, there was processing happening at every step. So we wouldn't have a connection in the brain or the brainstem if there weren't some kind of processing going on. But in some cases, we have to skip that processing. One of the best examples of this are those reflexes that we saw for something that's painful. Unless you're purposefully thinking about grabbing that hot bowl, a system for pain is designed where much of the thinking is done by the spinal cord. We don't worry about waiting for the brain to catch up. And so in the case of sound, in some cases, we have this fast track where some of the projections skip. That being said, for you all thinking about the pathway, you should stick to this kind of numbered pathway. I just want to point out that there's a line here and you might be saying, hey, what's going on with that little guy? That's what's going on with that little guy. The signals from the inferior colliculus are then relayed to a part of the thalamus called the medial geniculate nucleus. And some of these signals cross to the contralateral side. We're going to talk about a few different reasons why signals are needed on each side of the brain and some of the things that they do. But for the most part, they stay on this what is already the opposite side from where the ear that heard this sound was, right? Because we're passing now through from the auditory nerve, the cochlear nucleus, so on and so forth, all the way up to the medial geniculate nucleus on what is now the opposite side of the brain from where the ear heard the sound. And then the last place that this information goes off to for processing is onto the primary auditory cortex. And as we just said a minute ago, coming from the cochlea over here all the way through this pathway and up to the primary auditory cortex, this information is maintained in a manner that preserves this tonotopic map where certain frequencies are represented separately from others. So I know that that's a lot in a few slides. So what I did here, just for all of your benefit, for those of you that are taking notes, is that I put together kind of a quick summary. I think that might be really helpful. As you're trying to go back through and just, if you want to print out or maybe this slide will be on a separate page to where you can kind of look back through and you can look at all the different steps that are happening. But the thing you should remember from all of this is just the structures that this pathway goes through in order, right? We talked about a few things you need to know for the sensory systems, how transduction happens, what the pathways are, and then how we accomplish some of the functions. And so this portion we just talked about, and pretty much this quick summary here, is a list of what happens in the pathway to get from the ear to the brain. All right, now for some of the functions. So how do we localize sound? So how do we localize sound? We just talked about how sound is transduced. That's the way that we detect sound. And we said the next major function is localizing sound. And one of the primary ways that we are able to do this, even though there are kind of a few pieces that come together, is that we can localize sound by arrival time. So I think this will make intuitive sense to most of you. A sound that arrives from the left-hand side is going to hit the left ear before the right ear, and that creates a time of arrival difference. And this is very similar to the idea of the retinal disparity, where something might be a little closer to one eye than the other. Whereas sound waves coming from obviously the front are going to hit both ears simultaneously. And so if there's a big enough difference in where the sound is from one ear to the other, we can detect that difference in the arrival time. And the neurons that do this are in the superior olivary complex, and we find, much like some neurons are tuned to the retinal disparity, there are neurons in the superior olivary complex that are tuned to these spatial differences that occur in where a sound comes from. Now, there are actually a couple of different ways that we're able to do this. So we can localize differences in continuous sound, especially for low frequency sounds, by a phase difference. So the way that this happens is that if a sound were to come in from the front, then you would have both eardrums being pushed in at the same time, because the compressions and rarefactions would line up. However, when we have sounds coming from left to right, especially low frequency sounds that are going to have these kind of big broad sine waves like you saw in the crappy truck speaker system video, then you might have a difference in what we would call the phase. So one ear might be getting a compression and the other ear might be getting this expansion or rarefaction all at the same time. And so for low frequencies, we can detect that, where the two ears are kind of getting different pieces of that same series of vibrations that make up the sound wave. Part of the way that this coincidence detection works, especially we talked about these neurons in the superior olivary complex, is that there are sets of neurons that are tuned as what we call coincidence detectors. So the way I want you to think about this is almost like a series of dominoes. And so if I have sound coming in from the left ear, then it's like starting the dominoes out on this end. And if I have sound coming in from the right ear, it's like starting a set of dominoes on this end. And so when I have sound that hits both ears, it's gonna start toppling the dominoes from each direction. So when a sound comes in and it's closer to the left ear, we have these neurons that are basically built as coincidence detectors. And the only way that they're going to fire an action potential is if the two dominoes for the left and the right ear hit at the same spot. So if a sound comes in from the left ear, then it's going to have an advantage. It's going to start kind of tripping these dominoes a little faster. And so it might get to the fourth domino before it lines up with when the sound hit the right ear. And that would mean that only this neuron that says, hey, this is a little, this was a little closer to the left side, would be the one that would fire an action potential. In reality, this means we need kind of both signals to cause these neurons to fire an action potential. You can imagine the same thing coming in the opposite direction, where if it starts closer to the right ear, then we might knock down 1, 2, 3, 4, 5 dominoes, let's say, and then the sound finally hits the left ear, and then we kind of get these two dominoes to fall at the same time. This is where the toppling of the dominoes from both sides meets and kind of crashes or collides. And of course, the sound coming straight from the middle of the head is going to cause these to meet in the middle. And so that's the basic idea here, is that these coincidence detectors work by figuring out, by kind of passing the signal along until the two receptor potentials come in and meet at the same time. Now, one of the things that I said that's really, 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 really important is that for phase differences, we need low frequency sounds. And low is kind of a relative number. We've talked about low in a few different terms. But as sounds get to be higher and higher frequency, we're going to be able to do a poorer job of detecting these phase differences. And part of this is just because the frequencies are so fast that it would be impossible to even detect these separate compressions and rarefactions. However, there's a principle of high frequency sound that allows us to detect them in a different way, and that's using intensity differences, which is that really high pitch sounds decay faster than low frequency sounds. And you all inherently know this from some annoying roommate or neighbor or somebody that you've had once upon a time, because I think everyone in this world knows is almost a universal fact that when you have somebody that's hosting like a really annoying late night party or something like that a few doors away from you, um, you know, you never hear the high frequency sound. The thing that always carries through, which is almost more annoying, is the bass, and that's because low frequency sounds carry a whole lot further. But high frequency sounds are really susceptible to decay, and they're also more easily blocked by obstacles. And so this is where some of the features of the way that our ears and our head and everything are designed make a lot of sense. Because in our case, the head can act as an obstacle, and it can create what's called a sonic shadow. So if I have a high frequency sound coming from the right side of my head, then the head blocks some of that sound and it makes it reach the left ear as a sound that's a little bit quieter. So if the sound comes in from this side, then the head blocks it, and that means that the left ear gets a sound that's slightly quieter. And so these differences in the amplitude of the signal between the left and the right ear or some difference between the amplitude of the signal at the left and the right ear will occur, and our brain can also detect those differences in intensity. So this is the point where I bring you back to another really important thing that I raised like 100 slides ago, right? Which is, I said, well, why in the world do we have these ears that are shaped in like a crazy funny way, right? We've got these giant funky looking things that stick off the side of our head. Why in the world do we, have our ears instead of just a hole in the side of our head if all we need to do is detect sound? Well, another way in which we create a sonic shadow is through the ear itself. And so our ears, they're not just protrusions off the side of our head. I think one of the things you'll realize if you stop and take a look at yourself in a mirror for a tenth of a second is that our ears are pointed forward. So our ears are positioned to receive sound that's coming from in front of us. And actually the ear itself creates a bit of a sonic shadow when things come from behind us. So sounds coming from behind us are going to be quieter than ones coming from the front of us because our ears are positioned to be directional. And then for high frequency sounds, our head also acts as another way of creating a sonic shadow. So these are two examples of ways that we use intensity differences to help us locate sound. So we've kind of covered the main functions of sound, right? We know that for loudness, we would cause greater bending of the basilar membrane, and we can fire more action potentials. So we've talked about how transduction for different frequencies and also different loudnesses can happen, all based on just how much the basilar membrane moves. We talked about how we localize sound by using phase differences for low sound and intensity differences for high frequency sound. And then the last feature of sound that we talked about at the very beginning was timbre. And in a sense, you already know the answer to how we detect timbre, because what we've said is that the cochlea has a tonotopic map. So we can detect different frequencies at the same time because we can bend different parts of the cochlea at the same time, right? We can have a very complex sound come in, and because of the fluid in the cochlea being the primary mechanism for causing all these bends, then we can create bends in all kinds of different places that represent all kinds of different frequencies at the same time. So this is the first important piece for how we detect timbre, is we have a system that's designed to be able to simultaneously process all these different frequencies. And then the other thing that is important for processing kind of, complicated sounds and giving us some feel for what a sound is and where it's coming from is that information in the auditory system follows the same pathway as that in the visual system. And this is not some random coincidence. This is a very purposeful thing because we use that information from the visual system and the auditory system together, where information from this dorsal stream that tells us where a sound is and where a visual stimulus is are very likely going to be used together. right? Think about why these systems would have evolved in order to help with our survival. We localize a sound, we turn our eye to see where a predator is coming from, and we're very good at this. If you think about, the last time you were walking around in the woods and you saw, you heard a squirrel, rustle some leaves, your eyes immediately dart to almost the exact location of where a sound came from. So you can start to detect what's happening. And then the auditory system also has information that goes through the ventral stream that helps us to detect what. And so we start to put information together. And in some cases, we can use our audition alone in order to tell us what something is. Maybe you have spent all of your life, growing up in the Northeast, and you're very good at recognizing as you walk through the woods what the call of a robin sounds like. And so by the sound alone, you can do that. And maybe you're not so good at it, but you hear a sound of a bird and you turn your head using kind of your where information, and then you look and you see the bird and you combine the call together and you identify, this is a robin. So again, we put all these pieces of information together to give us some idea of where a complicated sound is coming from or what that sound might be representing using these locational features as well as the features we gain from all the frequencies that we call timbre. So as I said, but to highlight it here once more, these systems overlap because we use that information for all kinds of decision making, for navigating our world, and so these systems are parallel systems for a reason. So the last thing we're going to talk about, and only for a couple of slides here, really, is the vestibular system. The purpose of our vestibular system is to handle kind of a lot of different computations that are related to position, especially for our head. So one of the things that we're able to do with this is keep our balance. Our head in many ways is kind of the center of our world for balance and positioning. The other thing is that allows us to get some information about head position, and we kind of ignore the section of the chapter on vision about how we correct for movement of the head in vision and things like that, but part of that involves the vestibular system. So it gives us an idea of where our head is turning and how it's moving. And I say all that because I'm mentioning the third point here, where the information that comes from this system is what helps us to do that compensation, which for me is enough for you to know in this class and to walk out knowing the basics of how head movement affects all these things. One thing that I want to point out that I usually will talk about in the context of all of the sensory systems is that, we all think of ourselves as having 5 senses, but balance is not a sense that you would come up with, and yet we have a sense organ for it. And oftentimes the senses that we do think about, like touch, can actually be broken down into many subcategories where we have different receptors for things like hot and cold and pain and vibration and all kinds of stuff, as we'll see in another couple of classes here. So just keep in mind that, the brain's definition of senses is probably a little bit different than the elementary school definition for senses. So the vestibular system has 2 main components, the vestibular sacs and the semicircular canals. So the vestibular sacs respond to gravity as well as some information about head orientation. So these are kind of these little bulbs here. And then the semicircular canals, they respond to angular velocity. And so you can see that we have a semicircle that kind of goes in almost like an X direction. We have one that would go in kind of like a Y direction and another one in a Z direction. And actually, the mechanism for both the vestibular sacs as well as the semicircular canals is pretty much the same, where they both have a set of kind of really long cilia-like things that group together in what we call a cupula. And basically different kinds of movement will cause these cupula extensions to bend and cause then hair cells to bend. And this is what creates a receptor potential. So the semicircular canals are mostly responding to changes, right? So if you swiftly rotate your head from left to right, then that would cause kind of an angular movement of fluid through this semicircular canal that's on this x-axis. And if you nod your head up and down, it's going to cause fluid mainly to move through this one that's more in like this, this z direction, for example. And so we have that angular velocity change. And then, of course, if something were to knock you off balance, if you drop very quickly, then we have a similar response that happens in these vestibular sacs where they detect movement of fluid through the movement of these cupula and the hair cells. So that's a really, really kind of simplified version, but it's basically all I think that you need to know is that, the main point of the vestibular system is to act almost like the accelerometers in your phone, where when there's a twist, of your screen, then our brain knows to kind of change program because we've had a shift in our position in some way, shape, or form. And we use that information in all kinds of different ways, including telling us when we're falling or when our head has been twisted to one side very quickly and things like that. In terms of the pathway, there is a vestibular ganglion that is pretty much at the exact same spot as the cochlear ganglion. And really what it is that the cochlear and vestibular projections combine at some point in what some people call the cochlear nerve, but what most textbooks would actually call the vestibulocochlear nerve. So these group together on their way into the brain. And they pass through in the brainstem to the vestibular nuclei on the left or the right, or some of them, especially for things like basic balance, have direct projections into the cerebellum, which plays all kinds of important roles in movement and things like that. And then from the vestibular nucleus, there are projections out to a few places, but again, most of these are involved in things like correction of movement and stuff like that. So if you accidentally take a misstep and you activate the vestibular system because your body is now off balance compared to what it expects to be, we have direct projections from these vestibular nuclei to the cerebellum, to the spinal cord, to the medulla, and to the pons. All of these places are kind of brainstem or brainstem associated areas that allow us to directly make compensations for the change that has just happened to our body. So much less focus here, but I just want to give you the pathway because that's something we said you should know for each of these systems. And with that, I will just say you all know where to find me if you have any questions, and remind you that this material, for some, is the start of a more difficult set of material. So don't be afraid to come find me. If you have questions, if you've gone back through the lecture once or twice and you're not understanding things like how we localize sound, it's probably a good time to figure it out. My goal in this course is to quiz or test you on just the material that you need to know, just the core concepts, make certain you're walking out with those ideas, and really not much more. But it means that if you're having trouble with that, it's also a really, really great time to start asking some questions. All right, have a good rest of the week, and I will see you all next time.